Right now, 7 out of every 10 phase 2 clinical trials fail. Pharmaceutical companies spend billions of dollars and years of research only to watch their oncology drugs collapse at the finish line. The root of this 71% failure rate is the way we match patients to treatments. Today, clinical enrollment relies on a basic checklist of eligibility criteria: the patient's age, the tumor stage, or a single specific genetic mutation. This is static biology. It treats a cancer diagnosis like a photograph. We look at the tumor's mutations on day 1, assume that biological state is frozen in time, and prescribe a drug based on that snapshot. Cancer is a dynamic process. It mutates, adapts, and finds escape routes around our therapies. CrisPR-ai tracks these adaptive strategies using a framework called reasoning biology. To defeat a disease that constantly rewires itself, we have to move beyond observational snapshots. We need an engineering system that can predict the tumor's next move. This starts with the moment of diagnosis, using a newly diagnosed ovarian cancer patient as our example. Instead of checking if a specific gene is broken, the CrisPRO engine ingests the patient's complete raw genomic data to build a predictive digital twin. The system feeds this data into EVO2. EVO2 is a large-scale artificial intelligence model trained on over 9 trillion DNA base pairs to predict the functional impact of genetic variations. It reads the patient's genome to map out the current state of their biological pathways, like their DNA repair and immune response mechanisms. The output is a 7D mechanism vector. This is a mathematical profile that scores the tumor's specific biological vulnerabilities across 7 distinct dimensions, mapping exactly how the cancer is functioning. We now have a precise, quantifiable baseline of the tumor's weaknesses, replacing the guesswork of a generic checklist. With that baseline established, the system compares the patient's unique vector against a vast database of available clinical trials. This chart shows the mechanism-based matching logic in action. The engine mathematically compares the biological vulnerabilities of the tumor with the exact mechanism of the drug, aligning the therapy to the disease. And this operates entirely as a glass box. When a tumor board needs to know exactly why a drug was recommended, the system provides explicit data receipts, citation links, and pathway disruption numbers. They see the exact biological rationale. The patient starts the matched therapy and responds well, but beneath the surface, the cancer is quietly adapting to the drug. This graph shows the serial monitoring infrastructure. Over several months, it tracks the patient's blood tumor markers and pathway kinetics, watching for the slightest biological shift. If the tumor attempts to patch its vulnerabilities, the kill chain protocol activates. This is a strict multi-trigger system that cross-references different data signals to confirm the exact moment the cancer starts building resistance. Doctors receive an automated early warning, detecting the biological signs of resistance months before a new tumor would ever show up on a CT scan. Standard care often requires doctors to wait 6 months for a scan to confirm the cancer has returned, forcing the patient back into a cycle of trial and error. CrisPRO is anticipatory. The second the kill chain triggers, the engine automatically re-ranks all available treatment options to directly counter the tumor's newly developed resistance. It uses a feature called target lock. This designs specific interventions to intercept the tumor's escape route, blocking the pathway before the cancer can establish a new foothold. By identifying these escape routes before they expand, doctors can pivot to a secondary line of attack, aiming to stay ahead of the tumor's growth. When we scale this up from the individual patient to the entire industry, we address the $240 billion trial failure zone. This predictive architecture acts as a financial life raft for pharmaceutical companies. It ensures they only enroll patients whose underlying biology matches the drug, which improves the probability of a successful trial. Because the system also generates proactive pharmacogenomics, it identifies toxicity risks early, accelerating the regulatory pathway toward breakthrough therapy approvals. Oncology is shifting into a predictive engineering discipline. We can now anticipate a tumor's evolution and intercept it before it spreads.